Good evening, sir. Today I am present with Reactome analysis tools. Let's start. Introduction. Reactome is a free open sources curated and pre-reviewed knowledge based of biomolecular pathways. Its aim is to provide intuitive bioinformatics tools for the visualization, interpretation, and analysis of pathway knowledge to support basic research, genome analysis, modeling, system biology, and education. Reactome has grown to include entire for 8,701 human genes, reaching the 43% of the 20,296 predicted human protein coding gene, and 9,052 reactions annotated based on 20,708 literature reference, aiming to improve the user experience in terms of usability, responsiveness, and data visualization. All the major components of the Reactome web interface have been redesigned and re-implemented. Pathways overview. Pathways overview organized the hierarchically grouping detailed pathways like translation, protein folding, and post-translation modification into larger domain of biological function like protein metabolism. Pathways overview provide a bird's eye view of all the reaction pathways for the reactome, constitutes an entry point to reactome the progressively increasing the content for the human genome. Why are pathways important? The pathway response. The pathways are how cells work, going beyond the parts list into the mechanisms at the cellular level is a fundamental advance in knowledge. Solid response. Lots of omics database require data integration. Reactome is manually curated and peer-reviewed database of pathways and process in human biology. Open source and open access basic unit of Reactome. Reactome say extensively cross-referenced into infer the orthodox events in human species data expansion. Data exchange format from promote data integration, the analysis and visualization. psi midlab psi midlab representing interaction data representing interaction data networks. Biopacks biopacks used for the representing pathways and the network data. SBML representing modes of biochemical pathways, reaction, and the system biology network. Programmatic access. Application programming interface API. Biomart API are important to connect and automate data exchange between the local program and databases. Biomart API, PL API, Java API, SOAP and flat files, database dumps, local site install. Developer zone. The third parties can integrate the presented tools, pathway, browser analysis tools, recommend Reactome freebies, documentation, etc. Third parties integrating Reactome. Chemical entities of biological interest is a freely available dictionary of biomolecular entities focused on small chemical compounds. Integrates a diagram viewer to visualize the target reactions in the context of reactome pathways. Pathways diagram viewer. MI overlay. Molecular interaction overlay interactions data intact. The process during release and stored locally. Retrieved on a demand on the user request. Loading times improved to achieve a more responsive and interactive user experience. Include include an option to easily filter out the display the interactors by confidence score for the reactome. Protein and the chemical structures displayed for the closer view. Conclusion. Pathway visualization and analysis reactome offers a tool for visualizing and analyzing biological pathways. Researchers can explore the connectedness of genes, protein, and biomolecular genes, protein, and molecules in the specific pathway. In the specific pathway, helping to understand their roles in the various biological processes. Data integration. Reactome tools allow Reactome tools allow users to integrate their experimental data such as the gene expression, mutation, and interactions data with the pathway information. This integration helps the researcher gain the insights into how specific genes, how the molecules are the involved in the particular pathway and the processes. Enrichment analysis. Reactome provides the enrichment analysis tools that help the researcher identify how to identify them cancer genes or pathways that are the significantly enriched with their experimental data. Interactome analysis. The reactome tools can be used to analyze the protein-protein interactions network and the visualize how proteins interact within the various pathways. This is the particularly valuable for understanding protein the functions they are their roles in the cellular processes. The comparison and the cross species analysis. Researchers can be compare the pathway across different species using the reactome tools and enabling the study of the evolutionary uh, conservation divergence in the biological process. Reactome is a free open source uh, pathways database. They provide the molecule details of the biological network for the process in the context of the cell overall functional network. In the sense, it focused on the molecular events underlying the biological